for our last video for this chapter, we're going to get into neural communication and development. And so once we get through this, we'll be able to talk about conduction of nerve impulses, what is going to propagate that nerve impulse. So we'll get into the two differences, or the two major ways to do that in terms of saltatory and continuous conduction. And then we'll talk about the components of a synapse, and then look at electrical and chemical synapses. So this is the major thing. So we're going to talk a little bit about how neurons communicate, like how they send information down through themselves, but we also have to look at how does one neuron talk to another? Or how does a neuron talk to a muscle? Or how does it talk to a gland? This comes with the synapse. So inside the neuron, we're generating this electrical chemical impulse called an action potential that gets carried down the axon to the axon terminal. And at the end of the axon terminal, we have those little, remember I mentioned those little areas that are a little swollen, called a synaptic end bulb. And this is what's going to take the message from the neuron to the target cell, target organ, whatever. That we have the synapse, right? That is going to serve as the connection between the neuron and the target cell. Again, that target cell can be another neuron, it can be a skeletal muscle, it can be cardiac or smooth muscle, it can be glands, it can be any other part of the body, right? When we're looking at the synapse, we have the presynaptic cell, we have the postsynaptic cell. This is just talking about the order. The presynaptic cell is the neuron that's doing the talking. The postsynaptic cell is the one that's doing the listening, if we can kind of think of it that way. Okay, so looking at this, we see our presynaptic neuron, right? This is the one that would be taking that signal and sending it down the axon. And then these ones here are going to be the postsynaptic neurons because they're receiving that signal. So let's talk about the electrical synapses. So this is where a neuron and some other cell are going to form a physical connection through a gap junction. So a gap junction, we've seen that one before, right? That gap junction was one of the ways that we could connect one cell to another. This is going to allow an action potential to pass right through those channels. The electrical current is able to pass through to the target cell. We see these all over the place. We see them between excitable cells other than neurons, so like smooth muscle cells in the intestine, the cardiac muscle cells in the heart. They'll have these too, but this is a way that we can physically connect, right? So that the electrical signal can just go from one to the next without anything getting in the way. So here's that gap junction, right? So they're physically kind of locked into each other with these channels, and the action potentials, that electrochemical gradient can just go right through. The chemical synapses, this is the one you're probably thinking of, the one that you've learned about in a previous class. This is where we actually have a gap, right? So this is like I said, that little brother or sister who is going really close to you, but they're not touching you, and they're letting you know, I'm not touching you, I'm not touching you, right? Same kind of thing with the chemical synapse. So that neuron, the other cell, there's a little tiny space, a little gap in between them, and we call that gap the synaptic cleft. So there's no direct contact here, right? Instead, what we're going to do is we're going to send a chemical signal called a neurotransmitter. And there's a laundry list of neurotransmitters. We've already talked about one of them with the muscular system, which was acetylcholine, right? But that neurotransmitter is the messenger between the two cells. The neurotransmitter, like let's say acetylcholine, because that's the one we've talked about before, they're going to be packed up in little vesicles called synaptic vesicles. They're held at the end of the axon terminal, and when the action potential gets all the way down to the end there, the vesicles fuse with the membrane and basically spill those neurotransmitters out into that synaptic cleft. And then, since the gap is so small, the neurotransmitter can travel across that gap and bind to a receptor on the membrane of the postsynaptic cell. So the target cell, the muscle, the gland, the other neuron, whatever, right? And that can help generate a response in the target cell. So this is what we've seen before, because we saw this when we were looking at the neuromuscular junction with muscles, right? So that hopefully at least this picture isn't completely foreign to you because we've seen it before. But we have the presynaptic neuron. This is down at the end of the axon terminal. Right here's our little bulb. We have our neurotransmitters hanging out inside their vesicles. When that signal arrives at the end of the axon terminal, these vesicles fuse. They spill the neurotransmitters out into the gap, and some of those are going to get bound to these receptors, right, that are going to cause, in this case, this postsynaptic neuron to do something, right, to send another signal out, right, something. All right, for our last bit for this, so we can finish up chapter 11, we're getting into development. And so we're going to talk about growth and differentiation of the anterior neural tube. We're going to talk about the different stages of development of that anterior neural tube when it comes to developing the adult brain regions. We'll talk about the posterior neural tube, which becomes the adult spinal cord. And then we'll talk about the ventricular system, right, of the adult brain from the central canal of the neural tube. So if we remember, and this is like from a while back, so you might not remember, but hopefully you do. We talked about the endoderm, the mesoderm, and the ectoderm, and all the different tissues that come from each of those, right? Like muscles coming from mesoderm, and like your epidermis is coming from the ectoderm, right? But there's one that was kind of weird in that list, and that was the nervous tissue coming from the ectoderm, right? So the ectoderm, usually we think outside, so we're thinking skin, but it also gives way to the nervous system. The neuroectoderm is going to differentiate from the rest of it into what we call the neuroplate, and then fold into a neural tube. That neural tube is going to be what becomes your central nervous system. On the anterior side, it's going to become your brain. On the posterior side, it's going to become your spinal cord. Some of those cells are going to separate from the ectoderm to become what we call neural crest, and that's what's going to form your peripheral nervous system. All right, I know it's getting a little weird, but roll with me. So if we're looking at what's going on, right? Here's that neural fold and groove. We're forming that neural tube, right? We have this is the neural plate, right, coming from the ectoderm. So the ectoderm is what's in purple here, right? And then mesoderm and endoderm. So ectoderm is in purple. We see that folding in. That's creating the tube on the inside, the neural tube. And then the neural crest is what's coming here. And so that's going to end up being the nerves that branch off and allow you to communicate with like the muscles, allow you to communicate with receptors, like all that kind of stuff. So if we're looking at the neural tube, we're going to go through some enlargements, right? And so basically what we're seeing here is we're seeing like these swellings develop, and these swellings are going to become different parts of the brain. The first way we separate it out is the forebrain, the midbrain, and the hindbrain. Prosencephalon, mesencephalon, and rhombencephalon. I'm okay if you use forebrain, midbrain, and hindbrain. Then those become 5 secondary groups. So we're going to break down even more. So we had the forebrain, the midbrain, the hindbrain. Now we have the telencephalon, which becomes the cerebrum, the diencephalon, which becomes the thalamus, hypothalamus, the retina, which we'll see diencephalon again. That term's not going away, so you can't forget that one. The mesencephalon, which becomes the midbrain, the metencephalon, which becomes your pons and cerebellum, so part of your brainstem and the cerebellum in the back, and the myelencephalon, which becomes the medulla oblongata. So what we kind of see here, right, is like the hindbrain is starting to split in the metencephalon and the myelencephalon, right? And then we see the diencephalon developing as well. Those are like the new ones, the newbies in this. So this is what we're looking at, right? So here's this is what the brain looks like originally, right? It's not a whole lot to look at, right? So we just see they're kind of like these little swellings. And so we see the forebrain, the midbrain, the hindbrain, right? This is at like 3 to 4 weeks, right? A person probably doesn't even know they're pregnant at this point. And then that forebrain is going to split into 2, right? So we see what's going to become the cerebrum. we see the diencephalon, right? The midbrain, the mesencephalon, that's going to stay the same, and it's just going to become the midbrain. And then that bottom one, the rhombencephalon, is going to be the one that breaks into two to form the hindbrains, pons, cerebellum, and medulla oblongata. So if we're looking at, this is like looking at it head on, this is looking at from the side, this is looking at head on, this is from the side. So this is the beginning of the brain, right? What we're seeing on both of these sides of the slide. Not a whole lot to look at just yet. And then we have to develop the spinal cord too. So that posterior neural tube is going to be that long straight cord, little tiny hollow space down the center. That's going to become the spinal cord. So the dorsal tissues, so the tissues on the back of the posterior neural tube are going to be associated with sensory. The ventral tissues, right, the ones towards the belly, are going to be associated with motor functions. So we're seeing a split even within that neural tube of what tissues, which part's going to handle what in terms of what type of information. The neuraxis, so if we get into the neural tube, basically we're establishing like the anterior posterior dimension. This is called the neuraxis, so like the front and the back, right? So we have this anterior posterior dimension, and then we also have the superior inferior. So we're basically developing a top, a bottom, a front, and a back with the neuraxis. But there's this like weird curve that occurs between the brainstem and the forebrain. It's called the cephalic flexure. And so this is the weird part. The neuraxis will start in an inferior position, the end of the spinal cord, but it'll end in the anterior position, which is the front of the cerebrum. Right, but this is what we're doing here, is we're developing a top, a bottom, a front, and a back. Right, that's what we're doing with the human neuraxis, right? Top, bottom, front, back. We also have ventricles in there that are these open spaces, and this is where we have a bunch of cerebrospinal fluid like circulating and floating around in there. This is a remnant of that hollow center of the neural tube. So it's not, I don't want to say a leftover, because a leftover makes it sound like it doesn't do anything. And it's not that the ventricles don't do anything, because they're great for storing and housing that cerebrospinal fluid. But that's where it's coming from. It's coming from the hollow center of the neural tube. There's 4 ventricles and tubular spaces associated with that we can link back to that hollow center. Right, so 4 different ventricles. So if you're trying to kind of figure out what's going with what, this is a nice little table. Again, you can pause and look at this if you would like. But this is what part of the neural tube, what vesicle stage we get from it in the primary stage, what vesicle it's in the secondary stage, what adult structures do we get, and what ventricles do we get.